Hi, I'm Mark Gerstein, and I'm going to tell you a little bit about the research in the Gerstein lab. So overall, we work on AI modeling for multimodal biomedical data. And we do a range of different calculations, and we work on a whole variety of different data. So in terms of the data we work on one hand, very data intensive things looking at literature, EHR data, also looking at biosensors. And one of the key things we think about in this realm is sort of biomedical privacy. Um, the center of what we work on is relating these things to the genome and relating them to genetic variants. And here we're really doing human genome annotation and disease genomics. And one of the key ways we interpret the genome is through creating networks of genes and pathways for proteins. And we do a lot of sort of network analysis. Finally, we kind of drill into the molecules in a little more detail, often looking at their macromolecular structures in detail. We also look at pictures of molecules and pictures of other biomedical entities at many different scales. So we're doing a range of different calculations, and loosely we can think of the sort of first types of things. We're focusing more on data representation issues, data mining issues, and so forth. And the second range of things, we're looking more at a kind of elaborate simulation and modeling calculations. So I'm going to go into each of the areas in some detail. So first, let's focus on the uh, uh, disease genomics and the genome annotation first. So this slide just kind of gives an overview of the main thrusts in genome annotation. So on one hand, when people are annotating the genome, they're annotating it functionally. They have some assay that gives a kind of readout or signal over the genome, and that's what's shown here on the left. And then we take that raw signal and process it, make it more interpretable, and then we kind of create little annotation blocks from the signal. And we take these blocks and group them into bigger units, as I'll describe eventually, into networks. And the other thrust is comparative genomics, where we take the human genome and we look for conserved and similar regions in the genome. This can be just within the genome, where we're looking for duplicated regions in the genome, which might represent, for instance, duplicate genes or pseudogenes, or we can look within the human populations, particularly conserved regions or particularly variable regions. And finally, we can look between related species, such as human and mouse, again, looking for conserved or particularly variable regions. And again, the goal here is to find these kind of annotation blocks for conserved and very variable regions that we're going to use in annotation. Now, one of the key things we do is we take these annotation blocks and we group them into units to make networks. And this is kind of in the realm of what you might call network science. And here's a representation for kind of a molecular network. These are all the transcription factors in the human genome, and they're arranged in kind of a hierarchy so that the more kind of regulating ones sit at the top, and then the regulation you can see uh, flows downwards through arrows. And then we also look at how the regulation changes. For instance, this shows how the regulation changes in cancer, where it's kind of colored in terms of the rewiring in cancer. Now, we can take these network representations and use them as a kind of concise representation of a lot of genetic and functional genomic data. And here we're going to kind of make a model of those from those things. And so what we do is we actually take the network representation, for instance, the gene regulatory network, and we embed that network into some of the layers of a neural network model. So here it's a neural network model. And this particular neural network is going to take the genotypes at the bottom and predict something about the phenotype. In particular, it's going to predict something about um, mental disease, brain disease. And the neural network, the information is going to flow through it and highlight certain connections. And these connections actually are going to correspond, at least the bottom, to the nodes and edges in the gene regulatory network. So we can interpret the neural network and make sense of it. And this shows that particular process where we're going from a particular genotype to, say, predicted particular disease, schizophrenia, and we're highlighting certain genes, uh, regulatory elements such as enhancers and so forth that are important here. And this particular work, we've done a lot of this in relation to neurogenomics, and we've had it published in very prominent venues. This is the cover of Science a few years ago. Now, what are the big challenges going forward for a lot of genomic analysis? Well, one is just getting bigger and bigger genotype sets so we can get more power. Now, this isn't political power, it's statistical power. We can find associations. And one of the big blockades in getting more power is just getting people to consent to donate their samples to large-scale medical research. And one of the ways to do this is to make them feel comfortable with the privacy of this information. So we work a lot on privacy and consent. And then also another important aspect is even though we have lots of very precisely defined genotypes, we don't really have the phenotype that well defined. And so we've worked on defining the phenotype better. And so I'm going to talk about that a bit now. So here's an example of how we're doing neurogenomics, but trying to better um, describe the phenotype. And so what we use for the phenotype here is we take biosensors, things like just even a smartwatch and so forth, and we take the the time series data off that, and we process it in a special way by piping it through a neural network to get useful features, dynamic features from this biosensor data. And then we relate those features back to the genome in the framework of a traditional genome-wide association study, or a GWAS. And using the more precise digital phenotype, we can actually get stronger associations than we could otherwise. And this shows the associations we got actually for a particular brain condition called ADHD. And you can see we're getting fairly strong associations compared to a completely traditional GWAS. And then this vignette kind of talks about the issue of genomic privacy. So one of the tricky things in privacy is often we can link together different data sets and create these kind of quasi-identifiers that actually allow us to identify people in large-scale data sets. And this forms a type of information leakage. And we study this type of leakage from these linkage, what's called a linkage attack, in great detail and try to find ways of surmounting it. We also do a lot of work in genomics just looking at what's called variant impact. And here I'm showing you some of the work we did in cancer genomics, where we're looking at the many variants, the somatic variants you have in cancer, and trying to find the ones that have the greatest impact. And this is focusing particularly on the non-coding variants. And so here we prioritize these in a fairly simple way, looking at the ones that hit known non-coding annotations, ones that hit very conserved regions of the non-coding annotations, things that disrupt the non-coding annotations in an obvious way, such as breaking a motif, and that mess up molecular networks. We kind of prioritize them more highly. Also in cancer genomics, we've drilled in more detail at the actual genes and molecular structures, using the molecular structures to more clearly prioritize certain mutations, for instance, this driver mutations. And so this vignette shows how we can take an actual molecular structure and we can simulate it to find particularly intricate clefts on the structure, which could be the active parts of this protein, which are maybe more sensitive to mutations and maybe also more sensitive to drug binding. So overall, I've given you a quick whirlwind tour of the lab's research, and like I said, the focus is multimodal biomedical data and doing AI modeling on this. So here I've kind of showed the many different modes that we work on. We're focused very much on human genome annotation, disease genomics, but trying to relate this to um, biosensor analysis, EHR, literature mining, thinking about the privacy of this data. And we're also trying to drill into this a little bit more, grouping the bits in the genome into kind of more functional networks and pathways, and then looking at the structures in these networks and pathways in detail. And that sometimes involves some image analysis as well. And there's a whole range of calculations that we do that roughly fall on the spectrum from very data representation oriented type of calculations to data mining, deep learning, kind of machine learning calculations through to large scale simulation and very intricate modeling types of calculations. So I hope I've given you a kind of quick overview of the type of work that we're doing in the lab.